On this episode, Dr. Tom Cowan is here to share the New Biology Initiative and his quest to bring common sense to holistic family wellness. Quote, it is called New Biology because the, quote, old biology, based as it is on germ theory, genetic theory, cell theory, and a whole lot of other unproven or disproven hypotheses, is the wrong place to start a medical practice. Dr. Thomas Cowan is a well-known alternative medicine doctor, author, and speaker with a common sense holistic approach to health and wellness. Quote, we are launching our new biology clinic to give people access to the healing philosophies, strategies, and yes, even the details that I have come to in my 40 plus years in medicine. Tom has given countless lectures and workshops throughout the US on a variety of subjects in health and medicine and is the author of 7 books. Recent publications include Breaking the Spell, The Scientific Evidence for Ending the COVID Delusion, The Contagion Myth, co-authored by Sally Fallon Morrell, Cancer and the New Biology of Water and Human Heart, Cosmic Heart. Until his recent retirement from active practice, Dr. Cowan had a general medicine practice, first in upstate New York, and then for 17 years in Peterborough, New Hampshire, and for 17 years in good old San Francisco. Also, Tom was a founding board member of the Weston A. Price Foundation and currently serves as its vice president. Dr. Cowan continues to actively lecture and interview, sharing information via his website, drtomcowan.com. Dr. Cowan lives with his wife, Linda, on rural farmland in upstate New York. He has 3 children, 1 stepson, and 7 thriving grandchildren. Anyways, Bear, take it away.
Great. Tom, really looking forward to chatting with you. Always a pleasure, to have you here. So thanks for making time with us.
Thank you, guys.
I really Well, I consider you, one of the very important voices out there. You spent many years on the front lines actually learning how things function and then applying that to helping people. And I respect that very much. And what we'd really like to talk about today is your New Biology Initiative. So if you could fill us in on what's going on there, and then maybe we can use that along the way as a little bit of a launching place to go into some tangents about old biology and some of the things that doctor types might benefit in unlearning at this point. So welcome, Tom. Thanks again.
Thanks, guys. Great to be here with you. You're real pioneers and bright lights in this whole experience or whatever it is that we're all doing. I don't know, sometimes. So, all I can really say is when I look back on my life, I didn't know that there was a sort of defined, you might say, spiritual path that it looks like I've been on unwittingly or unknowingly. And that is, I think they call it in Hindu or something, the no path. And that path is essentially, don't worry about what's true, just find out what isn't true, and then somehow that process will lead you to the promised land. And there's often a choice here that we all make between like what everybody thinks versus what you can see with your own eyes or reason with your own mind, I think it is, or experience with your own heart. The argument that, well, everybody thinks you're wrong or everybody believes differently just doesn't work. So that led me, the heart doesn't pump the blood. There's a whole different reason the blood moves in the body. The, viruses don't exist, and cancer has nothing to do with genetics. And, sort of one thing after another. But what I discovered in the last few years is that process has taken like a kind of an exponential growth. And it was partly through actually conversations with Stefan Lanka, and then reading the work of Harold Hillman, and then reacquainting myself with Gilbert Ling, who's another biologist. And then I started to realize that this sort of debunking, or it's not this process, goes to just about everything in modern biology. There is no sodium potassium pump in the cell membrane. There are no receptors in the cell membrane. There's no such thing as an immune system. There's no synapses in nerves. There's no neurotransmitters don't promote nerve transmission. Genetics is mostly a bunch of hooey. DNA is not the stable element that determines the life or the structure of a living organism. And it just kept going. And I was not necessarily prepared or looking for this. It just was, well, then the next thing. And, you know, even something like yesterday, I actually did a webinar on malaria. I don't know if you guys have looked into malaria, but malaria is considered one of the crowning achievements and most settled scientific accomplishments of the last 100 years. Everybody knows it's this unicellular parasite called a plasmodium, and it's transmitted by mosquitoes, and mosquitoes bite you and then they bite the next person. And there's 5 stages in the life cycle, and that's why they do mosquito netting, and now they're going to roll out this vaccine and the whole bit. And, you know, as I presented yesterday, and I don't know that I want to go into the whole thing here, none of that's actually true. And in fact, when you go back to the 1880s and 1870s, when this was first being proposed, the people at the time, the doctors, the microbiologists, the toxicologists, the malaria people, said, these aren't parasites infecting your red blood cells. This is just what a red blood cell looks like when it's deteriorating. They've never seen these 5 stages. This one becomes this one, becomes this one. They've never pulled out a parasite, isolated a parasite, and shown that it caused disease in any human or animal. And even you have these crazy things like, you know, Haiti and the Dominican Republic are on the same place, right? Same island. Haiti has a huge amount of malaria. The Dominican Republic has essentially no malaria. So what is it? The mosquitoes can't fly from one place to another? I mean, you keep going into these things and you realize that none of the explanations essentially in conventional medicine, conventional biology, are actually founded in truth and logic and something called science. What I found out is, interestingly, it seems like there was a great reset in the 1850 to 1900 time frame, where before that, people didn't think there were germs, causing disease. They didn't think there was, DNA and genes and all this stuff that we think. They thought that we are basically an electromagnetic, entity that was living in the ether, which is an electromagnetic field. And we were tapping into that, and healing came about by like putting somebody in a cathedral that concentrated this electromagnetic field and actually stimulated a therapeutic response in the person. Or if you broke your bone, you would use sound or light and the bone would heal. And somehow in that time frame, we changed everything. We became a particle, right? We became a physical particle, or millions and trillions of particles, which obey random laws, right? And so I was essentially pushed by my son, and like, Dad, you got to tell people about this. You got to, tell people what's not true, and therefore what may be true, and then actually start treating them, but with these kind of guidelines. And that became the New Biology Clinic and the New Biology curriculum, essentially, exposing people first to what's not true, and then you wipe that slate clean, because if you don't wipe that slate clean, you go into this with all these misconceptions. So you get rid of all that. You realize what a beautiful experience we're all part of, but we just don't see it. And one of the things that I learned to ask myself with every single patient that came in, why does this experience you have right now, that fever or chills or sweat or whatever, why does this make sense to your body? And in fact, is the body's therapeutic response, right? Those symptoms, those are not the disease, right? In essentially, the disease model evaporates. There's no malaria, there's no, there's no AIDS, there's no cancer, there's no cold, there's none of that stuff. You're in a situation where something has happened, your body has to make it right, given the tools that it has and given the situation. And one of the things that I realized was so important with treating patients. And, this is a common saying in medicine, that there's no incurable diseases, there's only incurable patients. So one of the things, the people that, in a sense, scared me the most, scared me in the sense of I had the hardest time helping them. Like, so person comes in, so how you doing? I have fibromyalgia. So how do you feel? Well, I have fibromyalgia, and people with fibromyalgia feel tired and pain. So do you feel pain? When do you feel pain? Well, people with fibromyalgia, they feel pain at night and then sometime. And I couldn't get them to own the problem, that this is not fibromyalgia. I'm done with those kind of categorizing, or even malaria. Because if you look at the symptoms of malaria, it's, weakness, fever, sweating, chills. What are the symptoms of the flu? Weakness, fever, sweating, chill. What are the symptoms of COVID? Weakness, fever, sweating, chills. What are the symptoms of, lung moistures, fevers? And they say there's 247 million cases of malaria. And I always ask, are you sure it's not 246 million? And the reason I say it like that is because nobody measured, right? These are just wild guesses based on computer models. And second of all, there is no defined syndrome of AIDS, of COVID, of malaria. And so what you end up doing when you do realistic medicine is, how do you feel? What is your experience? Like, tell me what it's like to be you, and then how you got there.
This discussion is great. Could we use it to segue into genetic theories and things that you're coming to conclude at this point?
Absolutely. So let's put these things in. So what are we told? What are we taught? We're taught that that the blueprint of life is this chemical called DNA, which is found in the nucleus, and it's in a diffuse extended state normally, so that it can be transcribed, copied into mRNA in the nucleus. The mRNA goes out of the nucleus and goes to the ribosomes, which are in the cytoplasm, and it gets translated into proteins, and the proteins are our living structure, right? Does that sound right to you guys?
Yeah.
And inherent in that
high school biology to me.
High school biology. Every cell has the same DNA. You get half from mother, half from father, and it's unchanged through your life. That's your DNA. That's your blueprint. And you pass that on to your children. And this thing called evolution works because you get a change in your DNA through a mistake, or through radiation, or through chemicals, or just randomly. And some of them are worse and they cause disease. And some of them are better and they give you a selective advantage, and those are selected, and then your progeny are better off. That's the normal theory. So let's take a look at this, some of the details of this. And by the way, the DNA is a double helix. That's the Watson and Crick thing. Now, let's dissect that a little bit. So the first thing is the DNA is an extended form in the nucleus. It's a long strand, and then it gets wound up into chromosomes, which you see only when the cell is dividing. Now, I happen to be in college biology. There was a teacher there, a guy named Nichols, who I believe was eventually nominated for a Nobel Prize for discovering how DNA folds up into these chromosomes, right? So, and he was a brilliant teacher and et cetera. So you got this long strand, folds up into chromosomes. And I happen to get into his upper level biology class. So he gives a demonstration of how this works. So he has a string and he winds it around the classroom about 10 times and on like stanchions. And then he has a thimble and he says, okay, I'm going to fold this string and fold it into the thimble. And that would recreate how the DNA folds into forming these chromosomes. And he said that if this was a realistic model, there would be like 100 times the string and the thimble would be about this size. So he starts doing it. He's sticking the string and he's folding it back and forth. And he gets about 1.5 times around the classroom, and then no more string will fit in the thimble. And he starts with a stick, sticking it in there. And then he says, if after about 2 times, you can't get any more string in the thimble. Isn't it amazing that the living system, the body, can put this 100 times the string into the thimble? And everybody in the class stood up and cheered like, oh, the human body is amazing. It can do this. And I'm sitting there thinking to myself, wait a minute. He just showed me that doesn't fit into the thimble. And I don't think it fits in the thimble. And you don't have any idea how that string can fit in the thimble. And so I don't really think that chromosomes wound up DNA. So that was the first thing. The second thing is they have this theory that one gene, right, makes one protein. So that's how it works. So they do the Human Genome Project, 200,000 or so proteins, right? 20,000 genes. So there's 180,000 proteins that are unaccounted for by genes. So where's the code for them? Well, there isn't one. What do you mean there isn't one? That's the whole point, is that genes code for the proteins. You got 80%, 90% of them, there's not even a code there. Well, maybe they rearrange or something. I don't know how they come about. So that's a huge problem. We have 90% of the proteins, they don't even have a code. The third thing, so the DNA is made into mRNA in the nucleus, right? Then it goes out of the membrane of nucleus, has a pore. You can see that on a living cell. It's got a membrane. And you can measure the pH in the nucleus and in the cytoplasm, and it's different, which means the hydrogen ions can't equilibrate across the nuclear membrane, right? Otherwise, the pH would be the same. So how do you have a system where the mRNA made in the nucleus, which is thousands of times bigger than a hydrogen ion, can get out of the nucleus without letting the hydrogen ions get in and out themselves? It's the problem of how do you make a mosquito netting that keeps the mosquitoes, that lets elephants out, but keeps the mosquitoes from getting in? The answer is there isn't a mosquito netting like that. So there's no way for that system to work. Now, they realized that, and so then they made up the endoplasmic reticulum, which is a tube that opens into the nucleus. So the mRNA finds its way through the tube and crawls out down the endoplasmic reticulum. And then there's a whirligig in the endoplasmic reticulum, which binds to the mRNA, whirls around and deposits it in the cytoplasm, all the time not letting the hydrogen ions get in the whirligig. Now, when you go looking for a picture of the whirligig, there is no picture of the whirligig. So you say, how do you know there's a whirligig there? Well, if there was no whirligig, how would it get in and out? Right. So that's crazy because, it's like the sodium potassium pump. They measure sodium is different on the inside and the outside, potassium different inside and outside. There's a pump in the membrane with the whirligig that binds, and there's 2 Nobel Prizes for the sodium potassium membrane bound pump. So Gilbert Link comes along, strips the membrane off, and the sodium potassium concentration is the same, meaning there's no pump in the membrane that has anything to do with it, because if you take the membrane off, the concentration is the same. So they just made up a whirligig to explain what they couldn't explain. So that's another problem. That the fourth problem is it is absolutely definitively shown now that the DNA is different in every cell of your body or every tissue. There is no doubt, and it changes all the time. It's just one of the many chemicals, which I would say are just condensed frequencies, right? They're not even chemicals, really. They're just dense frequencies. And of course, they're changing and fluctuating. So there is no stable DNA, which is you. There's maybe characteristics like there is eye color, but there is no fixed chemical. That is a disproven hypothesis. So, I could go on, but there's so many, even, when I read Watson and Crick, how do you know that the DNA is a double helix, right? It's one of the foundational scientific discoveries of our time. I was just seeing if I had the paper. Nature, 1953, it says right in there. So a double helix is defined by the rotation, right? So every 10 base pairs, it rotates. And it says in there, we discovered the DNA is a double helix when we assumed that the base pairs rotated every 10 base pairs. And I remember reading that and thinking, what do you mean you assumed? That's like the whole thing. You're supposed to measure that, right? That's like 6 inches. I did a tape measure, it's 6 inches. No, they assumed it rotated like that, so that it rotated. I mean.
So, Tom, are they not? So I think a lot of people that are hearing this for the first time will say, well, can't they observe this with microscopes, slice a cell and see all this good stuff? I mean, I remember I was an honors biology student or AP. I memorized this stuff and they had all the models there. And you always thought, well, obviously this is just from direct observation.
Right. So DNA, it came from the famous Rosalind Franklin photo 51, which was an X. They took it, when you go to the quote methods section, how did they find this DNA? They took white blood cells, they extracted the nuclei, they mixed it with phosphoric acid instead of sulfuric acid. And then they came out with a phosphorus-rich chemical, which they said wasn't a protein because proteins are sulfuric acid. So this was a phosphorus-based chemical. It's something different. They called it nucleine, and that became DNA. They never seemed to think that the reason they got a phosphorus-rich chemical was because they changed the acid that they extracted it with to phosphoric acid instead of sulfuric acid. Anyways.
Sounds a little bit like virology.
it's all virology over and over. And then, so then they dry it and powder it. And then, believe it or not, and I have the whole thing right here. They expose this powdered chemical to X-rays for 61 straight hours, right? X-ray beam. This is the fragile chemical of life. Phosphoric acid, dehydration, centrifuging, X-ray for 61 hours, and they get an image on an X-ray film that looks like an X. And that became the visual evidence that it was a double helix. Now, the interesting thing about that is about 20 years ago, a bunch of grad students did a similar experiment, but instead they started with the spring of a ballpoint pen. And they got the identical image, which means that a reasonable conclusion is the blueprint of life is just as likely to be springs from a ballpoint pen as it is to be DNA. And maybe it is, I don't know, but I don't think so. So now, and when you say ribosomes, so you saw them in biology, that's where the mRNA is translated, made into proteins. There's thousands of these ribosomes. They're the factories of life. They convert mRNA into protein. They're hugely important with the mRNA shots because they go to the ribosomes. Now, here's the problem. And this, again, I get from Harold Hillman. When you go back, how do they find the ribosomes? What do they look like? They're all perfect circles on a two-dimensional electron microscopy image, every single one of them. You can't see it in a light microscope. They're too small. So in order for something to be a perfect circle on a dehydrated electron beam, chemically extracted, it would have been a sphere in real life, right? If you take an orange and you slice it longitudinally, and then you'd get a circle. So in other words, these ribosomes must be circles in real life. Now, what are the chances if you took an orange, like a circle, a sphere, and put it in a blender, that every piece would be a perfect circle? You know what the answer is? It's none. And that's what they did to the tissue to get this picture of a ribosome. In other words, it's an artifact. And Hillman proved it by taking something that couldn't possibly have a ribosome in it, and he did the whole electron microscopy technique, and he got the perfect circles because it's a gas bubble. And so somehow they showed you pictures of gas bubbles for the last 70 years, and there are no ribosomes. It makes no physical geometric sense or logic. There cannot be such a.
I will say, looking at dead stained slides or the electron microscopy has been one of the biggest downfalls of 20th century science. And I wish we could get back to like Gaston Naisson's style microscope or Royal Rife had to get the magnification so we could really start seeing the living terrain at a very microscopic level. And I mean, Royal Rife had this technology in the 20s. It's like, why can't look what we're supposedly have all this amazing tech. It's 2023, right? And yet we can't get a microscope like Royal Rife had in the 1920s.
Right. Yeah, I have a bit of a backstory on that where I was involved with some things I can't mention live here, but we were actually trying to fund the next level of microscopy because I actually did workshops with Gaston Naissons myself and, got to look in those things. And what happened is we were intervened by Interpol and FBI with the folks we were trying to create these technologies overseas with. So the short answer is certain interests don't want this information out. And, Mike, I told you, with the Bigglesons here, we're saying, hey, we've got all of these projects out there raising money that's not going anywhere. How about buying us a frickin' microscope so we can do what we already know how to do? So based on everything you just explained to us, which was fantastic for our audience, can you maybe present a more plausible theory on how cellular biology works?
Yeah. So if you just stick with reality, meaning what you can actually see and what you can prove exists, what you see is we have tissues. It's approximately 188 of them. 44 of them we know don't have any cells in them. They're syncytium, so they're basically organized acellular structures. The others may have cells, although I sort of doubt it. But let's just say they're organized tissue, or maybe even compartments called cells. And then you see this dome-shaped circular nucleus with a membrane. And then you have organized or coherent or gel-like water, which has protein infrastructure, like the scaffolding. And it also has lipids and minerals and other things dissolved in it. And then you have a very thin membrane around the cell, like Jell-O, has a kind of skin on it, but there's no real membrane. And then you have these inclusion bodies, which are called mitochondria. And they're like the power source, the power pack. They make ATP. That's at least the chemical carrier, but it's not to do with energy. ATP essentially binds to the proteins and unfolds them. And then the water collects on those unfolded proteins and forms a gel. The ATP plays the role that heat plays in making Jell-O, right? You put Jell-O, proteins, water, nothing happens. You heat it, unfolds the proteins, the water attaches, then you cool it and makes a gel. So that's what you have. You have, again, so you have this dome with an antenna in it. And then the DNA might be the antenna. And then it's embedded in a watery pool of crystalline, coherent, structured water. And then it's got a power source, scattered around in there. And that's it. Now, usually I ask people, and I can show you a picture of the Taj Mahal, right? And you see this dome with an antenna and then, capacitors into a pool of water. So it's the exact same structure, which tells you that that was not some tomb for some guy's wife or something. That was a power source. That was a creation of healing energy. It was harvesting the ether, the electromagnetic field, and bringing that information down into the water to create health and harmony and power for the people to use in their productive lives. And that's the same thing that happens in us. You have this antenna that downloads information from the light and the sound and the ether and your feelings and your thoughts and your intention. And just to say, consciousness in total. You harvest that energy, the dome acts like a capacitor, or it concentrates that energy field and it gets downloaded into water, and the intelligence of the water itself, no other code needed, allows the structures to be made that are needed by the organism, according to its intention. And that mitochondria just make the sort of power to sort of run the system. And that's it. And every illness and every natural therapy you can see based on that, some problem with that model. Like your water is distorted because you got poison grapes dissolved in your Jell-O. So what do? You heat the water up, that's the fever. You make it run out, that's called snot. You cough it out, and then you reconstitute a more perfect gel. That's illness. You have a faulty electromagnetic field because you think you should just have computers all day instead of the sunlight. You never got in the sun, and you never put your feet on the earth, or you're thinking toxic thoughts of I'm going to get people all the time and all that. That's what I do. then you download hateful information into your water, and you make, or here's a better example, you download lies into your water. And then your nervous system is a tangled mess. And what do we call a tangled mess of nerves? That's Alzheimer's disease. They say neurofibrillary tangles, which means it's all tangled up because yesterday you thought one thing and then you lied about that and then you lied about this, and it's all this tangled nonsense. So you can see the world gets downloaded, the consciousness gets downloaded into your physical structure. And that's why there's saunas and, cell salts. You're missing potassium chloride in the water, so the crystal isn't quite right. So you get a distorted protein, and then you have to get rid of that. And that's called a certain illness, unfortunately. And sweat lodges, hyperthermia, sound healing, light healing. It's all either the information or the receiver is off. And all you have to do is treat that and you'll be fine. For me, once you get the point here that this isn't what we were told, and that there's a whole nother, much more magical and beautiful and scientific way to see it, right? It's based on logic and experience and true experiments. It's just amazing what the kind of world that we live in. It's a gift to just be part of this.